be converted to a nervous impulse which can be conducted into the auditory centers of the brain, sound vibrations must pass through a number of ear components. If the vibrations are in the air, which is the case most of the time, these must be funneled by the pinna into the external acoustic meatus, which transmits them to the tympanic membrane. As the tympanic membrane moves back and forth, the vibrations are passed on to the malleus, incus, and stapes. As the stapes vibrates, the movement is directly transferred to the fluid called perilymph, which surrounds the delicate membranous labyrinth that comprises the internal ear. As the perilymphatic vibrations strike the outside of the membranous labyrinth, they are transmitted through its thin wall into the endolymph within the internal ear. Vibrations within the endolymph stimulate mechanical receptors called hair cells. The impulses generated by hair cells are conducted toward the auditory centers within the temporal lobes of the brain by the vestibulocochlear nerves. Did you hear all of that?
Hi, I'm Mike Shively, and welcome to Introduction to Anatomy and Physiology. We're currently talking about the ear as a spatial sensory organ. In a previous session, we talked about external ear and middle ear, and today we're going to concentrate on the internal ear and hearing equilibrium and that sort of thing. Did any of you have any questions from our previous session? Well, let's begin by going to the overhead camera. We defined osseous labyrinth last time, and I just want to start in at that same point. We said the osseous labyrinth was a set of bony channels, hollowed out channels, within the temporal bone. And within this osseous labyrinth, we have the components of the internal ear. Now, I've got a model. It's a small model. Got a bigger one here I could bring on. I'll just bring this bigger one on for just a second. And this basically is the shape of the osseous labyrinth. What I want you to notice about it is over here on this side we have a part that coils up, kind of like a snail shell, and it makes about 2.5 turns. That part of the osseous labyrinth is called the cochlea. Let me write that word up here. Oops. Let me write it up here correctly spelled. Cochlea. So that's this portion over here. Coils around about 2.5 times. Then that connects to a bulbous enlarged portion right in here, to which these 3 loop-like structures attach. That bulbous enlargement in this central area right here is called the vestibule. And these 3 loop-like structures are called the 3 semicircular canals. So all these are just subdivisions or particular parts of this osseous labyrinth. Now, none of the osseous labyrinth is considered to be a component of the internal ear per se, but it does surround and protect the internal ear. So cochlea, vestibule, 3 semicircular canals. These 3 semicircular canals are actually named. There's a posterior one, an anterior one, and a lateral one. It's not very important for us to know. But I do want you to notice that the arrangement, as I turn this model in various orientations, the arrangement of the 3 semicircular canals is roughly in perpendicular planes, such that we have one that's oriented more or less in a sagittal plane, vertically oriented in a sagittal plane, one that's vertically oriented in a coronal or frontal type plane, and one that is transversely oriented. So we have the 3 different semicircular canals. Now I want to really emphasize this nomenclature because it's confused a lot in the literature. Quite often we'll talk about the semicircular canals as part of the internal ear, and in reality they are simply bony channels that surround parts of the internal ear. Any questions about that before we start? Eva?
So is the osseous labyrinth actually a bone, or is it just kind of a space?
If you had imagined solid bone tissue within the temporal bone, go in with a little mini excavator and hollow out a series of channels within that look like this structure, the hollowed out area within the solid bone tissue is what we mean by osseous labyrinth. Think of it as like a series of interconnected cave tunnels, okay? All right. Another thing that I want to cover before we jump into internal ear per se is otitis. Let me write that word up here. I had a question about that after our last session. If itis means inflammation of, and OT is a word that means ear, this is inflammation of the ear. We commonly talk about otitis externa if the inflammation or problem is with the external ear, otitis media if the problem is middle ear, or otitis interna if the problem is internal ear. Otitis media, a classic example would be the situation that I talked about in a previous session where we have a collection of pus or fluid in the cavity of the middle ear, expressed as a pain syndrome because the pressure will push against the tympanic membrane. And of course, hearing acuity will be reduced simply because there's fluid surrounding the auditory ossicles and they're not free to vibrate, not as free to vibrate, nor is the tympanic membrane free to move. Otitis interna can be quite severe. My wife had a bad case of a viral infection involving her internal ear several years ago. And it only lasted a couple of days, fortunately, but during that time, her equilibrium was so interfered with by the infectious process that she couldn't actually walk. So it was quite debilitating, and of course there was a lot of nausea associated with that loss of equilibrium. Maybe you've had the experience of, well, you remember when we were little kids and we went out at recess, and I don't know what you did at recess, but you know, at my school, you know, you either played marbles or you did the little helicopter thing, you know, where everybody put out their arms like this and spun around, and the person who was standing, you know, after everybody else fell down from loss of equilibrium was considered to be the winner of the helicopter game that particular time. Of course, you had to learn not to do this particular game right after lunch because the nauseous center of the brain is very closely associated with the vestibular area of the brain, and so quite often you can get nauseous as well. Anyway, my wife had a bad case of otitis interna that really interfered with her ability to move. She had to kind of crawl from the bed to the bathroom, for example. It was that bad. All right, I want to make sure we had those kind of things defined before we started. Now I want to go back to this model that we looked at before, and I said that we can consider this to be a model of the osseous labyrinth, of those bony channels. But inside of those bony channels, we have the structures that make up the internal ear. Now, we defined internal ear last time as membranous labyrinth, a set of fluid-filled interconnecting channels. Now, if I turn this model up this way, the modelers have actually sculpted away part of the bone channel here so that we can see inside, and the gray-shaped tube in here is actually an internal ear structure. Now, the bony channel was called semicircular canal. The internal ear structure within it is called a semicircular duct. So within each of these three semicircular canals, there's a part of the internal ear called a semicircular duct. And the names are exactly the same except canal versus duct. So we have an anterior, a lateral, and a posterior semicircular duct. All of those ducts, in all three cases, connect down to a bulbous enlargement right here. Matter of fact, you can see two bulbous enlargements on this model. This one, to which the ducts connect, is called the utricle. And we'll spell that out in just a minute. I want to point these out on the model, then we'll write out the words. This bulbous enlargement in this location is called the saccule. So we have three semicircular ducts. We have a utricle and a saccule. And then within this part that we called the cochlea shortly ago, inside of that, there's a part of the internal ear that's actually called, let me hold this part up here so we can see it a little easier. It's actually called the cochlear duct. And it's got a couple of fluid-filled channels beside it that we'll look at on a larger model a little bit and see them a little bit better. So let me write the names of those parts of the internal ear out now. Well, first of all, let's go ahead and define. Let's go to a graphic and define internal ear as a set of interconnected fluid-filled tubes that are collectively called membranous labyrinth. I think we made this point last time. Internal ear and membranous labyrinth are interchangeable terms. Now let's go to the next graphic and look at the names written out of those components of the membranous labyrinth. We've got three loop-like semicircular ducts. They leave and return to that enlargement called the utricle. The utricle and saccule are those two bulbous enlargements. Those are found within the vestibule. And the cochlear duct is the part of the internal ear that is found within the cochlea. Adam.
So does the membranous labyrinth just sit right up against the osseous labyrinth, or?
No, as a matter of fact, there's a little tiny bit of space between the two. If we go back to this model on the overhead camera, I believe you can see this if we look very carefully. In gray here, we have the tube, in this case, a semicircular duct. And then can you see the pink color in there? That actually is a fluid that surrounds the semicircular duct. The name of that fluid is perilymph. So there's a little tiny bit of space between the limits of the osseous labyrinth and the membranous labyrinth within it. And that space is filled with the fluid called perilymph.
So the duct is kind of swimming in the canal?
You can, yeah. The duct, D-U-C-T, is swimming in the canal. It certainly is. As a matter of fact, there's a different kind of fluid inside the membranous labyrinth, too, and it's called endolymph. So we say that the membranous labyrinth, the components of the internal ear, contain endolymph, E-N-D-O, i.e. within, and then they are surrounded by perilymph, P-E-R-I lymph. Let me write those two words out and on the overhead camera here. The fluid that's found inside the internal ear is called endolymph. E-N-D-O meaning within. And the fluid that surrounds the internal ear structures is called perilymph, P-E-R-I-L-Y-M-P-H. P-E-R-I means around, within and around, if that helps. Perhaps, Adam, it might make this a little clearer. If I go back to this model, if we draw a transverse section right through this part of a semicircular canal, and let's just put it right down here, we'd have a circular structure and transverse section. And out here would be solid bone tissue. So this would all be temporal bone out here. And so that would be then the edge of the canal right there, semicircular canal. And within that would be the semicircular duct. I'm going to put a capital E in there to symbolize that the duct is a hollow tube filled with endolymph. And then between the duct and the canal, we have perilymph. So I'll put a capital P in there. So that kind of shows you the relationship. This space in here is filled with perilymph. And all parts of the internal ear basically have this same kind of relationship. We've got the bony tissue out here, temporal bone, and then inside that, immediately, there's a little bit of perilymph. Inside that are the internal ear structures, and inside those we have the fluid that we call endolymph. Does that help clarify that?
Absolutely.
Make it okay. Let's take a look at a graphic that summarizes what we just showed then. The fluid inside the internal ear is endolymph. The fluid surrounding the internal ear structures, exactly what you said, sandwiched between the membranous labyrinth and the osseous labyrinth, is perilymph. Now I want to talk about before we actually get down to the functions of the different components of the internal ear, I want to talk about the sensory receptor that's involved in converting motions of various kinds into sensory impulses. And that particular sensory receptor is called a hair cell. I want to draw a hair cell for us. We'll facsimile of a hair cell. So we'll go to the overhead camera here. And these are kind of columnar or tubular cells in shape. And they have an extension of the cell membrane that sticks off one end. They kind of remind me of like a little stick of dynamite or a candle, you know, with the wick sticking up. And the nucleus is down here someplace. And these are mechanoreceptors. Again, the name of this cell is a hair cell. Now, don't confuse that with cells found in hair follicles, you know, on our scalp or other parts of our body that produce hair. This is called a hair cell, and it's a sensory receptor that transduces movement into sensory impulse. If you move the hair of the hair cell, if you mess with this thing up here, it's like messing with the trigger of a set mousetrap. Move the hair of the hair cell, then this guy generates a sensory impulse. And depending on what part of the internal ear that sensory impulse comes from, that sensory impulse is going to be interpreted by the brain as either sound, i.e. hearing, or it could be interpreted as movement of some kind, either straight line movement or turning and twisting movement. Some parts of the internal ear have this particular function. Other parts of the internal ear are concerned with the interpretation of what we call sound. So hair cell, I'll talk about hair cells several times in reference to different parts of the internal ear, and there's some slight differences in the hair cell from one part of the internal ear to the next part of the internal ear, but the general concept is the same. Mechanoreceptors got this little process of cell membrane sticking out one end, and if you move that process, then the cell will generate an impulse, and the impulse, of course, is synaptically transferred to a sensory neuron, which carries that impulse into the brain by way of the vestibulocochlear or eighth cranial nerve. Now let's list the parts of the internal ear or membranous labyrinth and make sure that we can relate those to the kinds of sensations that we interpret from those parts. So going to the overhead camera, we have cochlear duct, which is the part that's coiled up within the cochlea. We have the three semicircular ducts. Which are the parts that are within each of the three semicircular canals. And finally, we have the utricle and the saccule, which is that pair of bulbous enlargements that are found within the vestibule of the osseous labyrinth. Now, the cochlear duct is the part of the internal ear that is responsible for what we call hearing. In other words, the sensory impulses that are generated by the cochlear duct, when those get to the brain, they result in audition or perception of sound. So cochlear duct, think of that as hearing. The three semicircular ducts, they generate impulses in response to turning and twisting kinds of motions. So when those impulses reach the brain, our interpretation is, hey, we turned to the left or right or torqued our head in some other direction. What physiologists call turning and twisting is angular acceleration. I'm going to write that down because you'll see it in your text. Angular acceleration. The utricle and saccule, when the sensory receptors within these parts of the internal ear generate impulses, the interpretation by the brain is straight line movements, forward or backward, up or down, left or right. And you know that even if you're blindfolded, you have that sensation. You can be blindfolded, get into an elevator, and you can tell whether the elevator went up or it went down, can you not? At least when it starts out, you can tell. Now, once you get moving at a constant speed, have you noticed that you can't tell really that you're even moving? You've had the sensation of being in an airplane, for example. And when you're taking off and landing, you know, that can be a little exciting there for a bit. But actually, when you're flying along at 570 miles an hour or whatever that particular liner cruises at, you don't have a feeling that you're moving at all, really, until you look out if you can see the ground, and you can certainly see things passing on by. At any rate, let me go back to the overhead camera and write what physiologists call this straight line motions that result from interpretation impulses here. They call that linear acceleration, straight line movement. Any question about this before we go on? What we're going to do then is consider each one of these separately and see how they do, what they do in order to generate impulses. All right? Let's look at some graphics that summarize what I wrote out. In the semicircular ducts, the hair cells, which are the sensory receptors therein, generate impulses when we turn or twist our head in the various planes that we can turn it or twist it. Next graphic, the sensory receptors in the utricle and saccule generate impulses in response to straight line motions, left or right, up or down, forward or backward, linear acceleration. And finally, next graphic shows that sensory impulses in the cochlear duct that are generated there result in interpretation of what we call hearing, audition, sound. Adam.
I did have a question really quick on the paralymph and the endolymph. Are those the same type of fluid, or are there differences between them?
When analyses have been done in the laboratory of paralymph and endolymph, they're very, very similar. They're water-based fluids with a relatively small number of other items within them. Paralymph was initially given that name because it surrounds, the structures of the internal ear around PERI. Actually, when they started doing some analyses of that fluid, they found out that it had the same analysis exactly as cerebrospinal fluid, and then later discovered that there's actually a very small channel that connects the paralymphatic space with the subarachnoid space, and in fact, paralymph is indeed cerebrospinal fluid. Endolymph, so actually paralymph then would be produced by the choroid plexuses in the ventricles of the brain. That would be its origin. Endolymph, found inside the structures of the internal ear, is very, very similar in composition, but it's actually produced by some specialized cells within the internal ear structure itself. All right, let's go back to the overhead camera for just a second and look at this model. I want you to notice at the base of each one of these semicircular canals, there's a bit of an enlargement, okay? And of course, the semicircular duct inside has an enlargement that's located in that position as well. What I want to do is I want to draw a transverse section through a semicircular duct located right where this enlargement occurs. Now, when we did this before, we said, here's the bony channel, and inside that is the membranous labyrinth, and we said there's endolymph in here and there's paralymph here. But if we go down to where that enlargement is, and I want to blow this up a little bit, we've got the bony channel out here, and here's the membranous labyrinth, the semicircular duct right here. Of course, there'd be paralymph out here, and there's endolymph in here. I'll put the P and the E in there to remind ourselves of fluids that are present. But where that enlargement is, there's a specialized structure that sticks out from the wall of the semicircular duct and goes almost all the way across. It's made out of a gelatinous material, and it's called a cupula. Embedded in the base of the cupula and sticking up into the gelatinous material are a bunch of those hair cells that we talked about. So I just drew some little marks down there and said those represent hair cells. Now, when we turn or twist our head in various planes, we get relative motion between this endolymph and the wall of this semicircular duct. When there is that kind of relative motion, this cupula, which is standing up like a pine tree in the wind, gets pushed by the fluid one direction or another. In other words, it bends slightly one direction or another in response to the fluid pushing up against one side of it or the other. And when the cupula is bent, the hairs of the hair cells down here are moved, and the hair cells generate impulses. So it's actually a bending of the cupula due to endolymph pressing against one side of it or the other that generates impulses. So the cupula is bent by the fluid, and impulses are generated in response. Now, if we go back to our model and take a look at it one more time, remember that we've got one of these semicircular canals and ducts in each of the three major planes: sagittal plane, frontal plane, transverse plane. So if we mime no with our head, which would be this kind of a motion right here, the duct within this semicircular canal that I'm pinching here between thumb and forefinger, there would be motion. You can imagine motion of the endolymph inside that particular channel. And so that would be the particular cupula that would get bent one direction or another. If we said yes, bob your head back and forth like you're nodding yes, okay? It would be this particular one. And if we basically took one ear to a shoulder and the other ear back to a shoulder and kind of wobbled our head back and forth from left to right, it would be the channels inside this in which the endolymph would be moving. Now, the brain unscrambles all this when the impulses reach it. So it knows which particular cupula from which particular semicircular duct was moved, and whether that cupula moved this way or that way in response to the pressing of the endolymph against it. Y'all know what a hula hoop is? Is that a toy that you remember?
Yep.
And you know that some of the hula hoops had marbles in them, you know. So, and you know that as you did the little motion with the hips, you know, I never quite got it down, but I tried a few times. At any rate, you can get that marble moving, okay? Well, once you get it moving pretty good, you can grab the hoop, and the marble's still going to zing around the hoop a bit, right? Does everybody have that experience? You've heard that and can imagine happening. Well, if we sit on a piano stool or bar stool and spin around, or doing that helicopter thing that I was talking about a while ago at recess, we can actually get the fluid in the lateral semicircular duct flowing kind of around in a circle. And then when we stop, the fluid itself does not immediately come to a stop. It continues to go around the circle for just a little bit. I don't know if you noticed, but these, let's go back to the overhead for just a second. If we can, to this overhead camera, we'll see that the three semicircular ducts all leave and come back to the utricle. So they're all interconnected to the utricle. They form continuous loops, if you will, through the utricle. So if we get that fluid in a semicircular duct moving, like we can by spinning around, then we stop, the fluid itself will still move. We will have the sensation that we're still moving because the cupula is still getting bent over. Hair cells are still generating impulses, even though we've really stopped. Okay? I've seen some machines that they have at county fairs. I don't know what they're called, but they've got 3 interconnected loops, and you can get yourself strapped in there. Have you ever seen those?
Been in one.
You have?
Yeah.
Well, tell me what your experiences were like.
You're pretty much all over the place. It's very disorienting.
Is it? Yeah. So do you have any trouble when you first get out of it? Are you?
Oh, yeah.
So you're messed up pretty good from what we're talking about here,
Yes.
Adam. Basically what you've done is gotten that endolymph spinning around in all kinds of planes, because those things will spin you in
all
every...
three axes.
Yeah. Any other questions or comments, Bryce?
Why does that affect the vision? I mean, you can feel it in your head, but also you see things moving as well.
Exactly. Matter of fact, if you watch somebody's eyes that is dizzy in this manner, you know, you'll see their eyes flickering back and forth. It's a nystagmus thing because the eye is assuming that you're spinning. So it's saying, well, I need to go this direction because the person is turning or twisting this direction. Its internal computer is messed up. It's trying to do what it thinks it's supposed to be doing. So.
Okay.
Seth?
Yeah, I remember going on a ride at Universal Studios Hollywood where it takes you kind of through this tunnel and the walls start spinning around. And before you realize the walls are spinning, you get this impression like the whole roller coaster is
turning
rotating,
upside down.
but it's not.
But it's not.
But you feel like you want to hold on to the sides and you kind of lean over and everyone is doing it. So can the brain mimic that just by seeing things?
Yes.
Spin?
Matter of
How does
fact,
that work?
visual stimuli can be very disorienting. I saw a movie once where individuals wore these specialized optical devices on their heads that made everything upside down and backwards, so that when it looked like you were, you know, when you're pouring water out of a pitcher into a glass, the perception through these glasses was that the pitcher was down here and the water was going up into the glass up here. Well, after these, they got some volunteers to wear this headgear for a few days, and it didn't take all that long to adjust to where it didn't bother the individuals because they were used to it, and the brain automatically flipped everything around in a logical manner until they took the headgear off. And once they took the headgear off and everything was back the way it was supposed to be, they were messed up again for a little bit, but then they adjusted very quickly. So it took them longer to adjust to the headgear than back, but nonetheless, the brain adjusted it and fixed everything. So kind of interesting, isn't it? Hazel?
Yeah, I was just wondering what vertigo means.
Vertigo means loss of equilibrium, dizziness. And so basically you can get that by doing the helicopter thing that we were talking about. All right, I want to go now to the utricle and saccule. Anyway, all 3 semicircular ducts work in that same manner. Let's go to the overhead camera and remind ourselves that these 2 gray bulbous enlargements right here are the utricle and the saccule. The utricle is the one that the 3 semicircular ducts come into and leave from, and then it is connected to the saccule. Now, they are primarily just fluid-filled bulbous enlargements, have endolymph in them, but they do have on one wall a specialized structure that I want to draw here on the overhead camera. The specialized structure is a gelatinous membrane called a statoconial membrane. I'm going to write M period for membrane. On the underneath side of that statoconial membrane, hair cells are embedded. So these little slashy lines represent hair cells. That's that specialized sensory receptor, mechanoreceptor that we talked about. And embedded on the surface, on the free surface of this statoconial membrane, there are some crystalline structures made of calcium carbonate complex to some protein. These used to be called otoliths. I'm going to write that word up here. And put quotes around it. This actually says ear rock when you translate the word roots here. The official name for these structures is statoconia. Calcium carbonate complex to some protein. So if this were the utricle, there's one of these things sitting kind of on the floor of the utricle, and the rest of the utricle is just bulbous space filled with endolymph. So we got this bulbous enlargement filled with endolymph, and the statoconial membrane with the hair cells and the statoconia sitting on the floor. You'll see, incidentally, in some physiology texts, the name for this whole complex right here is macula. A macula of the utricle and a macula of the saccule. Within the utricle, this sits on the floor of the utricle. It's actually physically attached to the bottom side of the utricle. And in response to straight line motions, forward or backward, or left or right, the hairs of those hair cells are shifted. You know, just like if you've got a book sitting on your lap and you're riding in a car with Mario Andretti, and he goes around the left-hand corner, you know, the book will shift what? To your right.
Right.
Okay? On the other hand, if he slams on the brakes, the book will tend to slide forward off of your lap. If he puts the pedal to the metal and takes off real fast, the book will slide back toward your tummy, right? You understand what we're talking about? Okay, go back to the overhead camera for just a second. And imagine this statoconial membrane sitting on the floor of the utricle. In response to forward or backward or left or right motions, then we get shifting or shearing motions relative to the hairs of these hair cells. And what do? They generate impulses. And so those impulses are carried then to the brain by the eighth cranial nerve, vestibulocochlear nerve, and the impulses are interpreted because they came from the utricle, and which hair cells are shifted which direction. Those impulses are interpreted as straight line motions, linear acceleration. Now, there's one of these same kind of structures within the saccule, but instead of being located on the floor of the saccule, it's located on the side wall. If we just pretend like this is the bulbous enlargement called the saccule, the statoconial membrane is over here in a vertical orientation with the hair cells coming in this side and the calcium carbonate crystals over here on this surface of it. And in this manner, because it's in a sagittal plane type format in a vertical orientation, then this particular statoconial membrane will generate impulses in response to up or down motions that will bend these hairs of these hair cells. And then also forward and backward motions will bend the hairs of the hair cells. So when that happens, then the brain interprets impulses from this particular sensory receptor as straight line motions of those kinds. So when we think of horizontal type straight line motions, think utricle. Forward, backward, left, or right in a horizontal type format. If you get in an elevator and go up or down, that straight line motion is detected as long as your head is, you're in the normal anatomic position. Everything changes, of course, if you're laying on a gurney in the elevator in the hospital, but nonetheless, that's the general idea that I want you to understand here. And why do we have these calcium carbonate crystals? Why are they there? What's the purpose of them? Anybody know? Eva?
Are they to add weight to make it so the
Add
membrane
some
will
weight or
move?
mass to that statoconial membrane to get it to shift appropriately in response to the gravitational or inertial forces that are put upon it. Evidently, they didn't work as well without the calcium carbonate crystals, so the creator added those maybe as an afterthought in order to give that membrane enough mass to adequately shift. For example, you're sitting there in the car, let's go back to our illustration, with a single piece of paper sitting on your lap, and somebody slams on the brakes, you know, there's not a significant shifting of that paper because it doesn't have that much mass. On the other hand, if you got, you know, volume A from the Encyclopedia Britannica sitting on your lap, somebody slams on the brakes, it's going to slide off your lap with considerable force. And so the purpose or functional role of those statoconia seems to be to add mass to that membrane. Bryce?
Why is it that some people they're able to move a lot more and a lot quicker without having any dizzying effects or anything than other people?
Boy, that's a tough one, Bryce. I honestly don't know that. I guess it's the same reason that I was going to be on the high school basketball team, but a coach, after watching me work out, said, you know, Mike, if you could dribble, pass, or shoot, we'd let you play, but, you know, there just seems to be some individual variation. I'm not sure what the why, you know, the answer to the why there is. Let's go now to the third part of the internal ear, and that's the cochlear duct. Now, I want to go back to this model on the overhead camera. Let me put it back together for just a moment. And this is the cochlea, the part of the osseous labyrinth. If we slice into it and look inside of it, we can see the components of the cochlear duct, which are actually this little triangular part. I want to bring a model of this particular area right up here, greatly enlarged, up for the overhead camera. And we'll probably have to back that camera off just a bit in order to, I don't know if we can get the whole model on there. We can get close. Yeah, and that's good enough. This is the cochlear duct right here. It's kind of triangular in transverse section, and of course the fluid that would be inside of it would be endolymph. Now this space here and this space here are filled with paralymph. This is called the scala vestibuli. It contains paralymph. And this paralymphatic space down here is called the scala tympani. Now I want to call your attention to the details of structure within the cochlear duct. This wall of the cochlear duct is formed by a very thin membrane called the vestibular membrane. Vestibular membrane. And that separates then the cochlear duct from the scala vestibuli. This membrane down here, which separates the cochlear duct from the scala tympani, is called the basilar membrane. And sitting on the basilar membrane, I think the camera can detect these, we may have to go in for a little bit of a close-up, are several hair cells. So these are hair cells. You can see the hairs, the little cytoplasmic processes sticking up, surrounded by their cell membrane, up against this membrane, which is called the tectorial membrane. Now let's see if we can go through that again. Vestibular membrane, basilar membrane, hair cells, tectorial membrane. So those are the main components of the structure here. Now, whenever we have sound vibrations in the air, those sound vibrations are going to strike our tympanic membranes. Tympanic membranes are going to vibrate. The malleus, incus, and stapes, the three auditory ossicles, are going to vibrate. And that base plate of the stapes, let me bring this back up. We'll pop the overhead camera on for just a second. We looked at this last time. This is the stapes, the stirrup, and it has kind of an oval-shaped base plate right here called the base plate of the stapes, and that fits in to an opening that's oval-shaped right here. It's called the vestibular window. Years ago, it used to be called the oval window. I kind of like that, but nonetheless, it's called the vestibular window. And as the stapes vibrates back and forth, it's glued into that window, but it has a freedom to move in response to sound vibrations. As it vibrates back and forth, it actually sets up hydrostatic shock waves in the paralymph. So if we go back to this model, we got paralymph up here, and we got paralymph down here, and because the stapes is vibrating, we get shock waves here and here. And those shock waves are transmitted right through this vestibular membrane into the endolymph of the internal ear. Because this fluid is vibrating, we get relative motion between the hairs of the hair cells and this tectorial membrane. And so these guys get bent a bit. And what do the hair cells do? Generate impulses. Those impulses then are synaptically transferred on to sensory neurons, which carry the impulses through the vestibulocochlear nerve to the temporal lobes of the cerebral hemispheres, where those impulses are interpreted as sound. So that's basically the structure: vestibular membrane, tectorial membrane, basilar membrane, and hair cells. Now, a while ago we were talking about endolymph and paralymph. There's actually some spatialized cells out in this wall of the cochlear duct in an area called the stria vascularis that actually slowly but surely secrete the endolymph. And I'm not sure how fast that stuff is turned over. Any questions there? Of course, out here on this model, this is all solid temporal bone out here. So the name of this bony channel is cochlea. That's the whole bony channel. But only this triangular part right here is internal ear. Question, Seth?
Yeah, what's the purpose of arranging the hearing mechanism in kind of that conical shape like that?
That's a good question. Why does this cochlea and cochlear duct within it spiral around 2.5 times? Let's take a look at this model one more time, if I can get him back together here. Why does it spiral around 2.5 times? Turns out that sound vibrations of different frequencies disturb hair cells at different levels on this spiral staircase. And so we've got, if the sound vibrations are very high frequency, they disturb hair cells right down here, and then maybe up here, and then maybe up here, and so forth. A different frequency will disturb hair cells at different levels. And that's basically how we determine different pitches. A high sound versus a low sound. I'm not talking about a loud sound versus a soft sound. I'm talking about a high note, a soprano type note versus a bass type note. And that's determined by pitch, or how many vibrations per second. Matter of fact, let's look at a graphic right now that summarizes that. When we talk about pitch of a sound, that's determined by the frequency of the sound vibrations, how many vibrations we've got per second. And the next graphic shows us loudness of sound. If we take a look at the next graphic, loudness of sound is determined by the amplitude of the sound vibrations. Other questions? That's a good one. Seth, Adam, you had one?
Yeah, if the stapes vibrating in the vestibular window sets up hydrostatic shock, where does that sound energy go? Does it just keep echoing back and forth?
Well, we've got a closed system, do we not? And it's filled with fluid, liquid, in fact. And liquids are relatively non-compressible. Now, you know, it's been a while since I've had physics, but as I recall, gases are fairly compressible. Liquids don't compress very well. That's the whole science of hydraulics, you know, and so forth, so on. But so we've got this. Go to the overhead camera for just a second. Let's take a look at this model one more time. So we got the base plate of the stapes fitting right in this vestibular window, and as it vibrates back and forth, we've got the fluid paralymph setting up shock waves in it. There, over here is another opening, and this is called the round window or cochlear window. Its old name was round window. You can see oval here, and you can see round here. You can see why I like those names. I can relate to that. Vestibular window over here now, and cochlear window. There's a thin membrane across this opening. It's called the secondary tympanic membrane. And whenever the stapes vibrates in and pushes on the paralymph in this location, this membrane over the cochlear window will bulge outward and thus allow that pressure to go someplace. So basically, we're not trying to compress the fluid, we're just moving the fluid a little bit within the osseous labyrinth. So every time the base plate of the stapes vibrates out over here, then this secondary tympanic membrane is sucked inward across the cochlear window. So that's how that works. And of course, these two openings, you realize that these two openings are into one wall of the tympanic cavity. So the auditory ossicles come across tympanic cavity, you know, and so the cochlear window is also in the tympanic cavity. So that these are free then to move. Any other questions? Bryce?
On the model, it's got that thing that comes out of the utricle and saccule.
Oh, yeah. Let's look one more time, the overhead camera. What is this structure sticking out right here? It's called the endolymphatic duct. It comes to a little blind-ended structure here called the endolymphatic sac. And we assume that this allows pressure changes to, we got a fluid-filled endolymph reservoir out here in this form of this endolymphatic sac. This simply allows pressure changes to either fluid to move this direction or this direction as necessary within the endolymph itself. So that's the purpose there. Any other questions? Hazel?
On that model you're holding, can you show us the vestibulocochlear nerve as
Oh,
well?
thanks. Yeah, we should look at that. It's shown here very nicely. These are various branches of the vestibular nerve. They're coming from semicircular ducts, saccule, utricle, as you can see. Each one of the semicircular ducts has a branch that comes in. The utricle has one, and the saccule has one. These all come into this part right in here. This is the vestibular nerve, and that's the vestibular ganglion right there. These are bipolar cells, incidentally. Down here, we have the cochlear nerve coming from the cochlear duct components. When these two things actually join together into a single entity that heads for the brain, this is called the vestibular cochlear nerve when we combine the two. So this is basically eighth cranial nerve and two different components of it. Okay? Other questions? Let's make a list of the structures that sound vibrations pass through all the way into where ultimately the impulses are interpreted in the brain. Okay? So what's the first thing? I'm talking right now, okay? And my vocal folds are making air molecules in this room vibrate. And what's the first thing those air molecules are going to interact with on you?
The auricle.
Going to interact with the auricle and external acoustic meatus, and they're going to be funneled into and strike what?
Tympanic membrane.
Tympanic membrane. So let's make a list here. We're going to say number one on our overhead camera. We'll list. We got sound vibrations in air. Got those vibrations in air. And then we got them in external ear. And then you said tympanic membrane. That's going to be, we're now moving to middle ear, aren't we? I'm going to write M-E-M period for membrane, okay? After the tympanic membrane, which auditory ossicle was it that's glued to the inside of the tympanic membrane? Malleus. So we're going to have the sound vibrations in the malleus. And the malleus articulates with what?
Incus.
The incus. And it articulates with stapes. Stapes articulates with the temporal bone, but when the stapes actually vibrates, those sound vibrations are next passed to paralymph. Then the sound vibrations have to pass through the vestibular membrane into endolymph within the cochlear duct. So these are the things that sound vibrations have to pass through before we get hair cells stimulated within the cochlear duct. And once we get those vibrations in the endolymph, then we get relative motion between the hairs of the hair cells, and the tectorial membrane, and the rest is history because they generate impulses. The impulses transmitted to the brain, and we get the sensation of sound. So I think it's interesting, though, to look at all the different structures that those sound vibrations have to pass through before we actually get impulses generated by the components of the internal ear. You know, I want to put some words up here because we didn't actually, I said them, but we didn't actually write them out. When we take a look at the components of the internal ear, let's go to the overhead for just a second. Had kind of a thick membrane down here, thin membrane here. This is going to be cochlear duct. I'm going to write CD in there for cochlear duct. Up here was scala vestibuli, and down here was scala tympani. I'm going to write S, V, and ST. But that membrane that came out right there, that the hair cells came up against, was called the tectorial membrane. Let me try to write that right here if I can get it all in. Tectorial. I didn't write that out for you. So that's that membrane. This membrane's easy because the hair cells sit on it. Here sit the hair cells. It's a basilar membrane. And this membrane was the vestibular membrane. I'll just write VEST, vestibular membrane. So those were the three membranes that we talked about. Kind of went through them real quickly when we were looking at that model of a transverse section. Remember, when you see this now, it's only this part right here that's kind of wedge or pizza pie slice shaped that is the cochlear duct. And this and this are both filled with perilymph and are not considered to be internal ear structures per se, but they do surround and are closely associated with the internal ear. And I see credits rolling, so I think we're good enough. We just have to not pick our nose and not do